Hi everybody, my name is Michael Sorochan Armstrong, and today I'm going to be presenting some of the work I've done with my colleague José Camacho on replicate node analyses for nested experimental designs in ANOVA-PARAFAC or PARAFASCA models. I work as a postdoctoral researcher in the Computational Data Science Lab in the Department of Signal Theory, Telematics and Communications at the University of Granada in southern Spain. So on this titular slide, what we have right here is a typical experimental design folded according to F1, factor 1, F2, factor 2, and then the associated variables with them. So if we're doing any sort of multivariate experiment with an experimental design, we're going to have a factor, and we're going to have the variables or measurements associated with that factor. And so if we're looking at factor 1, every voxel in this sort of, every voxel associated with factor 1 is a level in an ANOVA model, and as we sort of walk across this voxel along the variable mode, we have the multivariate measurements associated with that average level. So within this folded tensor are the average levels as predicted by a general linear model, which is related to the theory of ANOVA. And what we're looking to do is we're looking to sort of estimate the relative uncertainty with each level of our ANOVA model using these sort of replicate tensors that are folded similarly. But these replicate tensors, they will represent the uncertainty, and that could be the general linear model error, or that could be something else, as I'll get into. So sort of like I was talking about earlier, when we're doing a typical multivariate analysis, as would be the case for metabolomics or proteomics, a lot of the time we have a disease state that we're interested in, and we're looking at the difference between people who have this disease and people who don't have this disease, which may give us some insight into what the mechanism is for that disease within the body. And the nice thing about an experimental design is that if we're just doing like a regular PCA analysis, there could be a lot of confounding information that we're not interested in. And it's possible that the confounding information is related to variance for things we haven't really considered or wouldn't consider in a sort of typical classical principal components analysis. There could also be sex, gender, nationality, or occupation, and all of these things could actually have a much, much stronger influence on the metabolome than the disease state that we're interested in, because oftentimes the disease perturbation of the metabolome could be very subtle. So one of the first steps we will do in sort of separating these different experimental factors from each other is we're actually going to encode the different experimental levels according to something called sum encoding, right? And so with sum encoding, what we're looking at is we're looking at a way of representing this class information numerically. And how we do it is if we have three levels or three different classes within our experimental design for this one particular experimental factor, we can represent it as using basically two components, but representing it as a series of ones, zeros, and negative ones like this. So this gets into ANOVA-Simultaneous Component Analysis, and it's simultaneous component analysis, not principal component analysis, because it's a type of multi-block analysis. And the details of that aren't particularly important for this presentation, but basically what ANOVA-Simultaneous Component Analysis, or basically ASCA, does is it factorizes this experimental data according to our experimental design matrix. So that's, a series of our sort of sum encoding to do with the different experimental factors, right? And we do a general linear model. Basically, we regress this against that, and we get these coefficients. And then the reconstruction of these coefficients theta and the experimental design matrix give us this residual matrix that tells us something about the uncertainty with our model. And we can do significance testing using permutation tests by permuting this matrix and recalculating our statistics, right? And we can get a sense of, how statistically significant the different experimental factors are. And then we put it into a model. You can't really see it because my face is in the way. But each of these different letters here represent the sort of variance associated with a particular experimental factor, and they should all they're all orthogonal relative to each other, and we can also examine the interactions between them as well. So if the disease state is correlated with a specific occupation, say you're studying lung cancer and you notice that people who are welders and have higher incidences of lung cancer, that's something you could kind of look at. So ANOVA-Simultaneous Component Analysis, it goes in two steps. One is the significance testing step, like I alluded to earlier, and then the other one is the actual visualization. So after we sort of train this principal component model or a simultaneous component analysis model on the reconstructed matrices, as you can see by the scores and loadings, we take those loadings and we reproject the reconstructed matrix and the error matrix to recover these scores, which tell us something about the relative uncertainty for each level, for each factor and interaction. So in this example, the factor time between 24 and 72 hours for something else. Another sort of experiment we were looking at, you can see that there's some uncertainty with the samples associated with one measurement period versus the samples associated with another measurement period, right? And the same thing for the factor treatment, right? So you can see for the factor treatment that the levels are not very well differentiated, but they are for factor time. So PARAFASCA operates very similarly, right? You have this tensor that's trained on the GLM reconstruction, and then you can actually project an error tensor that's similarly folded to the GLM folding, because you need to fold it in order to perform the PARAFAC analysis. And you can use that to generate the relative uncertainty for each of the different experimental levels. And the nice thing about that is that we can typically visualize the same information using much fewer components, because the maximum rank of a PARAFAC model is going to be less than or equal to the maximal rank of a ASCA model. And typically the intuition is that PARAFAC models these things much more efficiently than principal component analysis does, because principal component analysis is going to fit the things that you may or may not be interested in, but by taking advantage of the multiple linearity in a PARAFASCA model, you should be able to visualize them much more efficiently. And especially as we're going to see for one component PARAFASCA models, it's often sufficient just to have one component versus maybe 12 components for complicated interaction. And that will allow you to communicate the findings of your experimental study with stakeholders, and it will be easier for you to visualize with less ambiguity in the interpretation. And that has to do with the uniqueness of the PARAFAC model versus the ambiguities present in principal component models. And just sort of getting to this, why did I say interactions could require more principal components? Well, interactions are the product, the sort of row-wise product between one factor and another factor. So if you have a experimental factor with 5 different levels, if you're studying 5 different diseases, and then you're looking at, another factor like 3 different occupations, right? You could end up with 10 principal components that you need in order to effectively model that. And analyzing every sort of pair or a triplet of principal components may get kind of difficult, because there's a lot of different combinations you'll need to look at, and not all of those combinations will be worth looking at necessarily. So what we did in an earlier paper that was actually a book chapter is we looked at the PARAFASCA, and we looked at projecting the error tensor back into the PARAFASCA model. And you can see with one component we have these error bars associated with each level that make it very clear what levels are statistically different from each other. In this case we didn't really do a significance test, but you can see that the error bars associated with these 2 levels are very well separated, and the first 3 or 4 error bars here might be well separated along the time axis, but you can see for the last 2 levels there's no clear differentiation. So our steps in the PARAFASCA model as we proposed it are: we model the unfolded data using parallel GLMs, sort of like an ANOVA step. Using the reconstructions, we train a PARAFAC model, then we project the error matrix as well as the factorized data into the PARAFAC model, and then we measure the difference in scores post hoc. So now we're going to get into a very special data set because it's a nested experimental design, and I'll talk a little bit about why the nested experimental design requires a little bit of extra care. So the paper we're going to be looking at is a paper that was written by my colleagues a couple of years ago, and it talks about an experiment that was done in Sierra La Baza, sort of in the south of Spain, and they were looking at the different trees, some of which had mistletoe, some of which didn't have mistletoe. They were looking at it across different seasons and at different heights in the canopy. And for each of those measurements, they were looking at a variety of different metabolite markers to tell them something about how mistletoe parasitism affects the metabolism of the host trees under a series of different experimental circumstances. So they were looking for confounding experimental factors that could be more significant than the mistletoe's effect on the host metabolome, because actually the mistletoe has a very sort of subtle effect when you're just looking at the tree. Obviously mistletoe is a different plant, but the tree does its best to sort of adjust to the risk associated with mistletoe to try and maintain homeostasis. So this is not a cross-experimental design, but when we're talking about error tensors, right, when we have our average GLM construction, we're constructing it on the average of each of the similar experimental levels. So here, this isn't a very good example because there's no identical experimental level, but if you're looking at HT1, you could potentially see that if there's another similar combination of these encodings, they would be averaged in the reconstruction, so they would be identical in the reconstruction, but the residuals that would represent the individual would be different and unique, and those would represent the uncertainty. So because of that, we're both interested in the average construction as well as the residual tensors, right? So we build a model with the average GLM reconstruction, and then we project the residual tensors back within the model. And yeah, it's very straightforward for a cross-experimental design, right? Because within a cross-experimental design, you're taking the same sort of individual and you're measuring it under two different circumstances, right? So in this case, we had T1, and its sort of equivalent experimental design is T4. Now, for a nested experimental design, that's what happens when we can't measure the same individual under two different experimental conditions, right? So in this case, the H and the P, so those are in this case representing the trees, the healthy and the parasitized trees. A better representation of this experimental design is like, look, tree 1 is healthy. We've only ever measured it when it's been healthy, whereas tree 4 has mistletoe on it. It's parasitized. So it can only ever be measured as a parasitized sample. So there are replicates, but it's not quite as obvious because when we create this nested experimental design, there's sort of similar information encoded with respect to this healthy or parasitized information here, but it's not, it is unique. Every level is unique with respect to the two experimental factors. So in this case, we would say that parasitism is nested within the tree factor. And this gets into something we're called hierarchical modeling, where the first order factors are parasitism, season, and height, and then the second ordered factors are actually the nested factor, the trees, the different trees we're looking at, the parasitism season factor, the parasitism height interaction, season and height interaction. All of these are on the second order. And why is it important what order they're on is because what they're measuring against during the statistical testing phase are these sort of first order factors. So as we do the analysis, what we find out is that with the exception of the tree factor, which we did not include here because we're not interested in it, we found that season and parasitism, as well as the interaction between season and parasitism, are significant. Height wasn't significant, and this, at first glance, it kind of conflicts with what our collaborators were finding, but something very, very important that we have to mention is that we actually did not include measurements to do with mistletoe. Just because mistletoe can't be healthy or parasitized because it is parasitized, right? So we weren't able to include that within our PARAFASCA model because it would introduce too much missing data, basically. So the results weren't exactly what they found, but we're interested in finding out how similar they are, how different they are, using a PARAFASCA analysis versus a more complicated analytical workflow. So these were the significant factors, and then within the hierarchical model we have the parasitism season interaction, parasitism and season as all being significant. And what we're going to do is we're going to try and project the residual information into the parasitism and season model and see what happens, right? So this is something that's really, really important to consider. This is our GLM model according to the experimental design with our tree factor, but because our tree factor is nested, we can't necessarily model the tree factor within the PARAFASCA model necessarily, because that sort of breaks the assumptions of trilinearity, right? If we're looking at the tree factor cannot be both parasitized and unparasitized at the same time, so it doesn't have that sort of multilinearity that we're looking for. So when we're folding the model, regardless of what factors we're incorporating, we don't have the tree factor, we don't fold with respect to the tree factor. So there's many different ways we can incorporate the error and the tree factor, because the tree factor is our replicate mode, but it's not necessarily an error mode because we've already accounted for it in the GLM model as a nested factor. So we could include it within the factorized tensor, and then the GLM error in the error tensor. We could include it within the factorized tensor and put the tree factor within the error tensor, or we could just take out the tree factor. So unsurprisingly, this sort of agnostic model did not tell us anything about whether or not the trees were parasitized or unparasitized. There was a very, very weak differentiation according to the height at which the sample was taken, but it wasn't really worth looking into. The most significant source of variance was, of course, the seasonal loadings, because the metabolism in the tree changes a lot between summer and winter. Unsurprisingly, in the conservative model, we didn't see any change in results, and that's because when you do not factorize with respect to the tree mode like we did in the previous example, it ends up in the error tensor anyways. So the results were pretty much exactly the same. In a greedy model, this is more interesting. In the greedy model, when we just factorize the trees out of both the reconstruction and the error tensor, we saw no difference between the two. So it's possible that the error in this model is quite high relative to the axes of the PARAFASCA model, and we don't really see what we're looking for in that case. Now, for what could be the best way of organizing the data, when we treat the nested factor as the residual matrix, so when we build our factorization and then project the nested factor as the residual matrix, we can see, just using one component, that the difference between unparasitized and parasitized is quite significant. We see no difference in the loadings with respect to height, and we still see the same sort of seasonal loadings with respect to whether or not the samples were collected in summer or the winter. And that agrees perfectly with what we saw in our GLM model, whereas the previous results did not agree very well with what we saw in our GLM model using just one component. And so while it's possible that we're overfitting in this case by using the nested factor as a replicate mode, so to speak, what you can see is that even in the most conservative regime, we can still see that differentiation according to parasitism status. It's just not quite as clear, and we need 3 PARAFASCA components to visualize that. Whereas when we visualize it using the nested factor, we only need 1 PARAFASCA component. So it's much easier to communicate a 1-factor PARAFASCA model with than it is a 3-factor PARAFASCA model. And another interesting thing about PARAFASCA is you can use this sort of replicate mode analysis to visualize the relative uncertainty with each of the different metabolites, and you can look for patterns in terms of metabolite classes with respect to that, and that's also very easy to interpret. So in conclusion, it is possible to model nested experimental designs using a replicate mode analysis in PARAFASCA, and we saw that with this data, organization of the significant factors and residuals does appear to alter the results of the post hoc testing. And we found that the replicate mode analysis using the nested experimental design best agreed with the general linear model that we performed in the first step. So with that, I'd like to thank the government of Spain, the Smart Eco Mountains program for supporting this research, as well as the Marie Skłodowska-Curie Actions postdoctoral program for sponsoring the MAHUD meta-analyses of heterogeneous data, for which this project is a part of, as well as Dr. José Camacho and Dr. Regino Zamora. Also, if anyone wants this wonderful cat, his name is Temperance. We found him on the street. Unfortunately, he has FIV, so he cannot stay with our cats who do not have FIV. But cats with FIV can actually live a very long and healthy life if they're given the proper care. So we're trying to find a good home for Temperance. Right now he's staying in our guest bedroom, but we really, really hope that somebody who's watching this will give him a good home. So thank you very much.